My great pleasure to welcome Dr. Krisztian Homicsko, who's an MD PhD at the Department of Oncology at the CHUV, University of Lausanne, is also working at the Swiss Institute of Bioinformatics. And we have two oncology projects here, and I think you're explaining everything about our oncology drivers.
Yes, correct. Well, I will, rather really speaking specifically of the SPHN project, I wanted to give you an overview about what we think in Lausanne about the computational oncology and molecular modeling, how we think that this could be used to develop novel strategies. And this project is federated by Olivier Michelin, who unfortunately cannot be here today. And so I'm trying to give you an overview what we think in Lausanne and what we think overall the SPHN can deliver to actually achieve a better care for our oncology patient. And in fact, responding to your question about personalized care, eventually this data can be then used for individual patients to decide and drive and direct therapy. So the precision oncology of oncology care is rapidly evolving. In fact, in the past, and not a long past time ago, we used to treat patients with standard chemotherapy when the response rates were fairly low. Then by understanding some of the biology, but mainly by clinical trials, actually, we figured out that some chemotherapies work specifically for some histologies. We still don't know why that would be definitely the case. And there we saw a little bit of increase of benefit of therapy. But with the advent of molecular profiling of patients, first we found, especially typically in, for example, lung cancer, subpopulations of patients with a particular mutation, which allowed us to actually define therapies with high response rates, but usually and typically in a very narrow subpopulation of patients. And of course, based on this data, one can imagine and project that we could actually find a unique set of therapies for every patient using omics data. And this, of course, will also increase the need for interaction between different partners, but also interactions between people, between specialists of their field to actually increase the number of patients to be able to narrow it down for a single patient. And so this is what is the paradigm of precision oncology. Although there are different disease types, as we learn over the times, different molecular changes, mutations, copy number variations, expression levels, subtypes of cancer, as well as immune phenotypes can be similar across different disease types. And understanding this heterogeneity of the population and narrowing it down for individual patients can actually help us to personalize the therapy. An additional layer of complexity, in addition to genomics, is actually the advent of immunology. And as well as in the genomic therapy, immuno-oncology can also be personalized. Initially, immune therapies were given to unselected patient populations, and we sort of saw some responses. Now we're starting to narrow it down for different histology, so similar to the histologies, patients with either with immune-deserted tumors, immune-excluded, or T-cell inflamed tumor. But the future is actually to understand, especially the resistant mechanisms that can limit the benefit from therapy. And then again, just as for the genomic therapy, individualized treatment for immuno-oncology treatments as well. And so there are already initiatives and ideas how to do that by creating a so-called immunogram, which can actually assess many aspects of an immune phenotype of a patient at a given time. But this can be extended beyond immune therapy also for the genomic status of a patient. So what we are looking for really and what we would like to identify are biomarkers. But given the heterogeneity and complexity of humans, actually usually these biomarkers are very complex. So they can be, of course, prognostic, where the actual intervention doesn't really have an impact on the outcome, but there is already a pre-existing biomarker, a pre-existing difference that defines the survival of the patients. The biomarkers can be predictive, in which case the intervention actually changes the course of the patients live longer. But most of the times actually these two are rarely really separate, and the markers are complex. They are both prognostic to some level as well as predictive. And so we are facing this complex and daunting task to identify this biomarker specifically to guide treatment decisions. And so what we want to do with precision oncology is for a given therapy to identify those patients who most of them or 100% of them will benefit from this therapy, but then exclude those patients who will not have benefit, then find a particular treatment that those patients, or at least a subpopulation of those patients, will benefit, and then give them that different treatment, and then find another treatment that will benefit then the additional different population of patients, so eventually achieving a high rate of response, and also, in case of immunotherapy, a lasting benefit. So what kind of data can we collect from oncology patients to personalized therapy? So of course, I will just shortly also go back to the clinical data because we see that this is fundamental to have high quality and good clinical data, environmental data, genomic data, immune histochemistry, pathology data, proteomics, metabolomics, molecular modeling. I will give you an example where I think molecular modeling could help in driving personalization, as well as the microbiome, which links very well to the environment as well. So our current working horse is genomic data. This is what we use the most frequently in the clinic, and this is what we would like to build on a network where the genomic data created at different centers can be integrated, analyzed to support clinical decision. Importantly, these biomarkers of genomic data are not stable, they are dynamic. So we have to take into account that, in fact, if a patient is being exposed to a therapy, for example, a first-line immuno-oncology or a first-line inhibitor therapy, then biomarkers for this setting would help define response rate or progression-free survival. But as the patients evolve and their disease evolve, actually, we need other markers. And in fact, the tumors will adapt. Cancer is a very adaptable disease, and we actually need other biomarkers, not just baseline, but also on treatment biomarkers and so on during the course of the disease to understand these changes and to alter the therapy accordingly. So what kind of uses we have for sequencing in molecular oncology? So first of all, we do already now in almost all Swiss centers covering large regions of genomes to identify somatic and germline mutations. Then in some instances, we actually go deep into identifying more rare variants of changes. Then we can also do, from a research perspective, larger populations of patients, doing, for example, whole genome GenVas analysis. It's important for us to do also to try to identify, using next-generation sequencing, the tumor heterogeneity. There's both population heterogeneity, so inter-patient heterogeneity, but intra-patient heterogeneity is a major headache for oncology and also a major driver of resistance. And as well as I just said, we use NGS to actually next-generation sequencing to follow up on patients, not just use a single marker at the beginning, but understand and adapt the treatment according to changes during the course of the disease. So we already have some evidence about personalizing therapy, oncological therapy, especially for kinase inhibitors. The first results are not that great, actually. So this is a study done in France, headed by Christophe Le Tourneau from Institut Curie. This was a French study, randomized. They included 741 patients. They could give matched therapy to 195 and unmatched to the other 50% of the patients, and they see no difference. They had no benefit from the treatment. Another example from MD Anderson is somewhat better. They actually included 500 patients. They could match 188 patients, either with the therapy or an unmatched therapy. And here there is an improvement both on response rate, progression-free survival, and overall survival between matched and unmatched patients. And then this is a recent study from the Moscato group, which highlights many of the difficulties of such studies. They screened 1,000 patients. They found 411 actionable mutations in 843 patients. And then out of these 840 patients who did have a molecular profile, they only could treat 199 patients. And out of those 199 patients, actually only 22, 11% of the treated 194 had actually a benefit. Overall, actually, it's less than 5% of the patients who actually derive benefit from the 1,000 patients. But when those patients did derive benefit, they actually could measure the progression-free survival before and after matched therapy, and there was an improvement by matching the therapy. But one of the challenges is actually finding actionable mutations and actioning on those mutations. So this also highlights the problem when the action is happening, that there are other factors that can define difficulties to respond. And so I just want to give you an overview of a specific change in melanoma, which is one of the diseases we are treating with Olivier Michelin, and what can we learn from targeting the MAP kinase pathway, which is particularly modified in melanoma. So selective inhibitors are around for 10 years. They produce spectacular responses. So this is all the black knots here are melanoma. And after a few months, the disease disappears. But then eventually, another 6 to 12 months, the disease comes back. So BRAF is a clear oncogenic driver in melanoma. And the BRAF mutation is very early even. So this is why these melanoma cells are dependent. But Another 50% of melanomas don't have any BRAF mutation, so those patients cannot benefit from this spectacular treatment. And in addition, we know that it's not only BRAF mutation that these patients harbor, but they have plenty other mutations which we are ignorant about and we cannot and will not impact on. So, for example, again, in human melanoma, we see a heterogeneous populations. Only 50% of the patients have these specific BRAF mutations. Another 25-30% of the patients have another kind of mutation, NRAS, which is not targetable, although we have some drugs that they don't work. Then we have another population of patients, again, still in the MET kinase pathway called neurofibromatosis 1 mutant, NF1. Again, they don't really have any therapy, and there is a group of patients who have no benefit, no target at all identified. And so we can give more or less to two groups of patients some drugs. These ones really work very well. This one is questionable. But again, we do nothing about the other alterations. Do these alterations have an impact? And in fact, we now know that it is the case, and it's even treatment specific. So we can identify changes in the PI3 kinase pathway, which is a very well-known pathway, again, in melanoma and other tumors. And we see that these changes, either in PTEN or in the PI kinase pathway, they actually have an impact on the response rate or on the progression-free survival, and they are actually mechanisms of resistance. Interestingly, whether we do monotherapy or combination therapies, we actually see a different effect. So it's not just that we identify resistant mechanisms, they have to be seen in the context of a specific treatment. Another problem is the equality of mutations. So not all mutations of the same protein are the same. So here, just give you an example of a protein MAC1, and then 4 different mutations. And what you can see, this is a bona fide mutation which actually activates the kinase domain of MAC. There are 2 other mutations. So there's another mutation here, which is P124Q, which actually does the very same effect, opening up the kinase domain and making it constitutively active. But mutations are at the very same position of 124, which are L and S, don't have the same effect. They do not destabilize and do not activate. So 2 different mutations can have the same effect, and mutations at the same position can have also a very different impact on the function of a protein. And this needs to be understood in order to personalize the therapy. And so now we are moving away from a single gene, single therapy paradigm and actually to a model systems biology approach, because we understand that not a single gene is capable of defining and predicting the response to a therapy. So this requires a lot of network analysis, both for the genes, but also for the participating members of the networking group. And so here we come to the SPHN and also the collaboration part, where we would like to integrate data. And so one of the things that we just discussed, that the genomic data is relatively straightforwardly produced. The mutations calling can be complicated, but it's also quite similar. What we see as a major hurdle, at least in oncology, is really the quality of the clinical data. And so there's a big effort now to create databases, to create data that is structured, that is easily interpretable in order to actually be comparable between the different centers. We are setting up minimal data sets, which is still in the process to define what is the minimal data set. It should include diagnostic, structured clinical staging, structured treatment lines, structured evaluation of responses. Then the molecular data has to be comparable between the different centers. We're actually looking and trying to understand, are there any additional parameters that need to be defined, toxicities, body weight, other factors that can define, in order to then feed in together to be able to actually extract data and compute novel variables. And so the data strategy, this is our local data strategy, but we're trying to combine and compare it with other centers as well. It's based on 3 different elements: existing data, structured data, and we're moving most of the existing data, so in prospective way, to a structured database, and then as well as still existing free text from which we need to extract data. So what kind of existing data we already have? We have the diagnostics we need to transform. We have the treatment information. We already have response, which is quite difficult to read because it's most of the time the radiologists are not really trained to report them in a structured way. And then we have difficulties, but we also have solutions to collect long-term survival data. So one of the things that we realized was really difficult is actually having evaluation, which can be different between different centers. Now there are standardized evaluation ways for imaging. And we, in Lausanne, in CHUV, we actually convinced the radiologists to report systematically in a way that can actually be used and be comparable between our one center, but also potentially with other centers. As you can see, clinical trial patients have 100% go through this process. Non-clinical trial patients, as this is going forward, are actually a little bit lagging behind, but we're trying to catch up that every patient who goes through goes through the oncology department gets a precise evaluation according to given criteria. Structured data, again, we use one data set, a database system software, Sorian. It allows us to actually structure the data properly, and then we can use it for data mining. And I just wanted to give you an example how this data mining can help. In fact, we try to look into the patient's dossier written by doctors to identify the different treatment lines. It was really difficult. It is almost impossible. But when you use the structured data, and notably billing data for the patients who went through the hospital, we can actually very nicely develop what we call the patient view of the different treatments that the patient received according to the time, as well as whether the patient at a given time point developed the complication for a given treatment. This also helps us to evaluate the progression-free survival, all the changes that happen to the patient's course disease. So initially in Lausanne and in the Roman network, what we have seen there are these two different clusters, one Geneva, Lausanne, and the other one in the Swiss-German part. So we were part of initiating a Roman network of oncology, which build on this data and try to actually create this system. So what we do still in Lausanne and Geneva is we reach out to the local hospitals where the hospitals provide us with blocks and slides in order to perform omics data, which is pretty much the next generation sequencing. Then we also gather patient electronic records. It's being sometimes difficult, we actually have a flying data manager who collects the data to make sure that we have all the information needed. Then we perform the data mining, which is interpretation of the data, and then we propose treatments to the patients in one case. In another case, we actually propose clinical trials at the two centers where patients can come in, also from peripheral or private care, and then once the clinical trial is finished, we refer them back into their initial doctor. And so this works very well. We also initiated and implemented a molecular tumor board, which now we're trying to extend beyond the Roman network to whole of Switzerland, all the university hospitals, to have a common language and a common way of discussing and presenting patients. And so this is what was initially just a local effort, and now it goes into beyond. One of the SPHN driver projects, SPHN project, Swiss Personalized Oncology Network, which includes almost all the university hospitals which do oncology care, as well as the Swiss Society of Clinical Cancer Research. And so together we initiating a way to harmonize the data. So the long-term goal, of course, is of the Swiss Personalized Oncology Project is to define treatments for an individual patient. But actually where we stand right now, the short-term objective is really building interoperability. We see that there are huge divergences between the different centers, both in interpretation and action on the clinical and molecular data. And especially the clinical data is a very important headache to be interoperable. And so these are all the members of the SPHN network, and so we have frequent meetings to discuss the progress in this project. And now what I would like to just in the second half of my talk and the last time of my talk, just give you some example what we think we could use the data generated in the SPHN using still machine learning. There are some advantage of machine learning. So this is one example when we did serum proteomic analysis of melanoma patients, and actually using machine learning algorithms, we could identify biomarkers or a set of biomarkers that can efficiently separate patients. We use different methods. I will not go into the detail. We used the variable important analysis as well as the minimum depth analysis, and we found that these two different mechanisms based on forest plot could actually they've correlated very well. So we found gene or proteins that correlated equally aided by the minimal depth or the WIMP analysis. And what is the importance, and we really think this can be used in the future, that we can build relatively simple decision trees based on a multiplex marker. So here we analyzed 400 proteins, but this data can be extended to many more other parameters. And then this is a very robust way, so we can identify responders and non-responders to the immune therapy for a particular immune therapy, but not for another kind of immune therapy. Another example is where we think the future will go is understanding intratumoral heterogeneity. This is what we do is single-cell RNA sequencing. So this is another kind of next-generation sequencing going beyond the genomic understanding. This is a patient's tumor where we can readily identify using single-cell sequencing based on 4,000 cells, the different populations, which is, of course, very important in immuno-oncology. We can then quantify them according to the different types of cells. We can also identify heterogeneity within the group. So here, melanoma cells are splitting into two heterogeneous groups as well as the myeloid cells. We can also identify biomarkers. So this is PD-L1 and PD-1 expression on different clusters. And they actually correlate very well with what we see with just pathological analysis, and they are very important in decision of immuno-oncology therapies. But in addition, we can actually see pathways. We can compute pathways within the cell types. So here, ERK, AKT, or Wnt pathway. And then we can also see the impact of treatment. So here, the before and after treatment, we see the shift in these different cell populations. And then to finish up, actually, we think that one of the important elements is molecular modeling. We see the problem by analyzing the data of individual patients that beyond classical mutations, which are easy to, which are nicely listed, there are lots of mutations of unknown significance that have not been cataloged in terms of functional, non-functional, and inhibitory ways. So what we do with molecular modeling is predict the impact of a given mutation that has not been listed before, whether it's oncogenic, pathological, or whether it can be therapeutically targeted. And so together with Vincent Zoete and Olivier Mücken, there's a development of a Swiss PO, which is a web-based tool to predict mutation pathogenicity. And so this is based on our minimal panel of 52 genes. One can query. This will very soon go online. You can select different mutations and then query whether that particular mutation is predicted to be pathogenic, and it can be targeted or not. And there's a very easy platform also to do the structure of that particular residue, as well as the evolutionary maintenance or importance of that particular residue. And the next step is actually we would like to use machine learning algorithms, but our major problem is that most of the machine learning algorithms currently are difficult to interpret. So this is in collaboration with the Swiss Data Science Center. We are implementing interpretable machine learning algorithms, which can actually be probably more simple, but also more interpretable for the human. And this will be fed back into the SPHN project as well as to other research projects. And so in conclusion, we think there are clinical grade multiomics are rapidly available already, but these data formats will need to be complemented with well-captured clinical variables. This is really a prominent problem. Data integration will require many experts. We still think machine learning will be crucial, especially if they can be interpreted. Thank you.
Giving us this update on the latest of personalizing oncology treatments. Maybe time for one question. Peter?
Thank you very much. For this wonderful talk. I admire your optimism because you touched a little bit the dynamics of these tumors. They can mutate again and again and again, and as you have seen, the mutations increase and increase, and we are always running behind. Do you think we will be successful with immunotherapy, for instance? That's my first question. The second, immunotherapy has a lot of side effects. I mean, if you do it, for instance, with the CAR T cells technology, then these antibodies can attack normal cells, and then you got a lot of inflammation reactions and a lot of other side effects. So the quality of life is probably miserable.
Correct. So the two questions there. One, well, I work with George Kukos, so I have to be optimistic about immunotherapy. So we think that it is going in the right direction. We see across disease types the responses, and also those patients who don't respond, they have a particular reason why they would not respond. And by modeling animal models and other, we can actually understand and modify that lack of response. So this is one thing. And second, well, you're absolutely right. The side effects are bad. And many of the patients don't need the therapy, still get the side effects. This is what is is one way of biomarker, pre-select the patients for immune therapy, for one kind of immune therapy or another, and only expose those ones to the very high toxicity who really will get the benefit. So this is one way of reducing the exposure. The other, actually, we have a sub-study of predicting immune side effects, and it is possible to predict immune side effects, and some of that it has to do with life experiences of patients. And so by understanding and you need a lot of data for that. So human population, one is a genomic heterogeneity, but there is all the rest: environmental, microbiomics, origin, race. So all of that will have an impact on the eventual side effects as well as the response. And so we think that eventually you can reduce the side effects and can avoid, but I'm treating melanoma patients with Ipi/Nivo, and we are fine with the side effects. But there are some which are cumbersome, which are less forever. But other than that, patients are ready to take the risk. So we'll see. But it is not that much a hurdle.
Thank you. I think we'll now have a coffee break on the agenda, and I'll ask you to be back on 10 past.